So we'll start immediately with the science. So first, just one slide about why we want to do single-cell RNA-Seq. And the motivation for us to do this thing is because we want to understand the heterogeneity of complex tissues like the brain. And the problem is that, of course, this is very complicated. This tissue is very heterogeneous, and it's heterogeneous in many terms. It's heterogeneous, for example, in the morphology, in the size of the cells, in the activity, in the connectivity, in many aspects. And this is just one example of tissue, a very complicated tissue that we want to study. Now, before we start to discuss the technical details, just two basic questions. And the first one is, why do we want to do RNA-Seq? And the short answer is that basically RNA-Seq is a systematic readout. And today, with the technology we have, we can measure it in a very high quality. We can measure it genome-wide. We can measure it with high specificity and high sensitivity. And this is all due to the specificity of DNA binding. And the hypothesis that lay behind this kind of measurement is that the transcriptome as a whole contains enough information about all these cellular properties that I just discussed, that the tissue is heterogeneous, like morphology, electrical activity, and enzymatic activity, and other properties. Now, the second question is that you want me to ask himself before diving into this challenge is why doing single cell? And just as an analogy, I put this fruit basket, and to study this mix of fruits, what we used to do is, in the tissue level, is basically take all this mix of fruits, and in order to study the component and all the properties of this, put everything into one blender, mix everything, and then try to answer questions about the property of these foods. But I think quite a long time ago, people figured out that it's actually very difficult to answer questions about particular elements of this mix. And because we basically average out everything, and if the mix is heterogeneous, we basically lose all the signal. Instead, and another idea is to take each one of these fruits and put it in individual blender, and then analyze the composition of this fruit, and then we can basically answer particular questions about each one of these elements of the fruits. And what we want to do is basically the same with cells. We want to take a very complicated tissue like the brain, dissociate it into single cell suspension, and then put each cell in a different compartment and analyze its composition. So this is basically the motivation and a little bit of background. Now I will move directly and briefly explain a little bit of the molecular biology behind this, the single-cell protocols. So basically, when we say RNA-Seq, we always actually do cDNA-Seq. And when we talk about the normal protocol, we start with basically cells. We break them. We get the RNA. This is called lysis step. Then we use reverse transcription that basically copy the RNA into DNA strand. This is called the first strand synthesis. Now, in bulk experiments, basically we have many cells. Sometimes people start with millions of cells. We lyse them. We get tons of RNA. We can reverse transcribe it and get a lot of cDNA. Then it's easy. We can immediately continue into standard library prep into sequencing. But in the case of single cell, of course, what we have is a single cell, and that's the whole idea. And what happens is that we end up, after the reverse transcription step, with very little material. So what we need to do from practical reasons, in order to still handle these samples and measure them, we need to amplify this material. And basically, people use, there's two main ways to do it. One is with in vitro transcription, and one is with PCR. And the protocol I will discuss today uses PCR to do this amplification. Now, one of the main key features of this protocol that I'm describing for single-cell RNA sequencing is the idea of tagging the molecules before we amplify them. So remember, again, the first step is basically similar. We break the cells, we get the RNA, and then we reverse transcribe it. But now I will zoom in into one particular RNA molecule, and we mostly work with polyadenylated RNA, and we'll explain the process. So in the mix, we have reverse transcription primer, which is poly-T primer with a known tail on the 5'. Then what happened is that the reverse transcriptase started to copy the RNA, and when it gets to the end, this family of reverse transcriptase enzymes, MMLV, usually add, when he gets to the end of the molecule, he tend to add 3 nucleotides, and they are with high tendency CCC. In the same mix, what we usually have is auxiliary oligo that we call template switch oligo, that just we add to the reaction in high concentration. And since it's high concentration, and since we have on the 3' side of this oligo GGG, this oligo will tend to hybridize to the CCC, and then the reverse transcriptase enzyme will continue copy into the same first strand cDNA, this same oligo that we call template switch oligo. And this whole process called the template switch. And what we get at the end is, if we forget now about the RNA strand and we just look at the cDNA, what we get is basically we have the cDNA, which is the copy of the RNA molecule. We have the UMI, which is something we add using this template switch oligo, which is a set of 6 random nucleotides that will be the tag, the unique tag to this specific molecule after reverse transcription. And most important, we have a handle how to amplify this molecule now, because we have on this side and on this side, both on the 5' side and on the 3' side of this molecule, we have known sequence. So now we can come with primers and amplify it. So this is the next step. Basically, we can use a single primer PCR because this primer and this primer have the same sequence, and amplify this material, which again, remember, it's a single cell pool of RNA molecule or copy of the RNA molecule into a large amount of material that we call the single cell cDNA library. And this is the amplified material. Now, why do we need this to add this tag that we call unique molecular identifier, or UMI? So after reverse transcription, again, the molecule looks like this. We have the mRNA, we have the handles, the primers, and we have this UMI. But now, since we use PCR, you can imagine that if we have a large set of molecules that are illustrated by this small number of molecules, after PCR, what happens is that the PCR amplification is not perfectly uniform, and thus each molecule may be amplified with different efficiency. So what we get is that different molecules will be represented differently in the amplified material. And this is not so great, because what we are interested is to quantify what was the pool of molecules before the PCR. But this is actually why we have the UMI. So now when we take this pool of amplified cDNA molecules and we sequence them, what we get is we get reads, and then we analyze those reads. And what we can see is that 8 reads, for example, came from this molecule, 3 reads came from this molecule, etc., etc. But actually now we are able to go back from the pool of the amplified material to the pool that was before the amplification, and basically say, no, it doesn't matter how many reads, it matters how many molecules, and each one of these is actually one molecule. Yeah? And now I'm talking about molecules, I'm not talking about genes. It can be that this molecule and this molecule represent the same gene. Again, to illustrate this, we can think about this example. Again, it's 2 cells. They express similar genes, but with different abundance. So if we focus, for example, on the blue molecules in cell 1, we have 4 molecules, while in cell 2 we have only 2 blue molecules. So we make reverse lysis, reverse transcription, and the amplification, again, the amplification is not perfect, so we amplify it, and we get the PCR. And then remember that the blue molecule represents a single gene. But if you look at the labeling, this blue molecule got the green tag, while this blue molecule got a gray tag. And here again, each blue molecule got a different tag. So now we amplify it, and then we put together all the blue molecules. So again, if I look at cell 1, what I see, again, I don't look at how many reads, I look at how many different molecules, and in cell 2. So the final quantification that I want to make is that in cell 1 we had 4 molecules, while in cell 2 we had 2 molecules. And this again represents what we are interested in is the quantification of the initial state, the amount of molecules we had in the original cell. And again, this is something that we can do only due to the introducing the UMI before the amplification step. Now, the last step of the protocol is to take this amplified material, the cDNA, from a single cell, and to prepare it to Illumina sequencing. And also, of course, we don't want to run a single cell on the whole lane. We want to run hundreds of cells on single lane. And for this, we need to add another barcode, which will enable us to multiplex the cells. So we do this library preparation of this protocol using TN5. And this is basically similar to the Illumina Nextera protocol. So we start with, again, relatively large amount, nanograms of single-cell cDNA. And this is how it looks after the amplification. Again, I'm looking now in a single molecule, so it's a double strand now. Again, we have one side with the poly-T and one side with the UMI. We are interested to sequence only the 5' in this protocol, because the 5' contained information about the UMI, and it contains enough information about the gene, because we can read the beginning of the molecule, and then it's enough in order to align it to the transcriptome and say which gene is it. So now we basically load the TN5 with oligos that contain the Illumina adapter. So we have one Illumina adapter on this side, on the 5' side, and we need to add the second Illumina adapter. So this is basically inside these oligos that the TN5 is loaded with. The TN5 is a transposon. It will basically cut the DNA and introduce those oligos. Then we have a step of isolating the 5' of these molecules, and this is how the final final molecules in the library for sequencing will look like. On the 5' side, it's the same thing. We have the primer, which is the Illumina adapter. We have the UMI. We have the 5' side of the gene, of the cDNA. And on the other side, we have basically the second Illumina adapter and what we call a cell barcode, which is just there in order to allow us to multiplex many cells on the same Illumina lane. So this is basically the last step of in terms of the molecular biology, and now this material is ready for sequencing. Now, to perform this protocol, we started to use about 3 years ago the Fluidigm system. Very briefly, the Fluidigm system is a commercial microfluidics platform that allow you to perform basically cell capture and all the lysis, reverse transcription, and PCR on chip in very tiny chambers that are all controlled with a valve system. And this is a very beautiful engineering, and the volumes of the reaction makes everything very efficient reactions. So this is an advantage of this platform. The second nice advantage of this platform is that you can image every cell. So we can basically, after we capture the cells on this microfluidics chip, we can image each individual cell, and what you see here is only the zoom in into the exact capture site, and we can produce this report. You can see the cells on this capture site, and you can, if you look at the cells that I particularly highlight, you can see that different cells have different morphologies and different sizes, and we can quantify those cells. And we can also know where there are cases that it's not a single cell, or it's not a cell at all, or it's a dead cell. And then we can basically eliminate those cells and avoid to analyze them. And this is very important in single cell analysis to avoid all cases of doublets. So this is basically the second nice advantage of this platform. And today, of course, it's a little bit important to say that the main disadvantage of this platform is that it's a low throughput, so you can capture only 96 cells on a single run in this kind of chips. Now I'm going to briefly go into the last step after you acquire this kind of data from large enough number of cells, and you have a nice data set. So the next step you want to do is to analyze this data. And I'm going to describe briefly the approach we took. And again, I will say it up front that there is an explosion of methods, and I will just describe one method that we use in the papers that we publish in the last 2 years. So the general theme in this in analysis of basically single cell experiment is that we want to look at heterogeneity, and basically the theme is that we want to take this full data set, which is genome-wide. So what you get is a big matrix. You have genes on the rows and samples and cells on the columns. And now what you want to do, basically you want to get clusters or groups of co-regulated or groups of similar cells. And we call them clusters in this analysis. And basically they are potentially de novo cell types or cell states. And the standard, this is not related to the Bextin protocol that I'm going to describe, quite general. The first step you want to do in such analysis is to do a feature selection. Feature selection means to reduce the data set from all the genes that describe each cell into a small set of genes that are the most relevant, that contain enough information but do not contain a noise. And this is standardly in single cell. I think today many people use this kind of approach, basically plotting the log of the CV versus the log of the mean, fit a line, and then select for the top genes that are above this line, this fitted line, as the highly informative genes. And again, this is not based on standard just variance filter, but on this fit of the CV versus mean approach. After you did the feature selection, you get a reduced data set. So means that you go from 20,000 genes into whatever, the top 1,000 genes or 3,000 genes. And then you can start basically doing your analysis. And the goal is to take this reduced data set and create something that is clusters. So something that you can look at, basically grouping the similar cells together and the similar genes together. Then you can start to look at the data. The procedure we took is we call it Bextin, mainly because it's based on the spin algorithm. Spin algorithm briefly is an algorithm to sort distance matrices or correlation matrices into one-dimensional order. So it's not a clustering algorithm, but it's a sorting algorithm. Developed by Eitan Domanic's lab about more than 10 years ago. Basically, the steps that this Bextin take is basically the idea is to do binary split, to take this data, divide it into two groups that are really separable, and then repeat the same process on the subgroups, and then continue doing these splits until we don't have enough information to split the cells. Practically, if we look at the first split, we take the data, we sort it into a one-dimensional order of the genes and the samples. And again, this means that we use the spin algorithm, and the spin algorithm will sort the cells, for example, and it will put cells that are close to each other in this one-dimensional order. And this is very important because it reduces the problem of how to split the data into two chunks into a much more simple problem, because now we have only the number of possibilities is reduced to the number of cells. And this is everything here is done on the correlation matrix in our case. So after we sort the matrix, the correlation matrix, it has usually some structure, like is illustrated here. And this is illustrated, I'm just illustrating the correlation matrix of, for example, cells by cells. And usually, if you look at those, the blue squares represent clusters of cells. And now the task that is not part of the spin algorithm is to find the optimal breakpoint into this correlation matrix. And for this, I developed some heuristics how to do this. And basically, you can appreciate that those kind of points that are between the squares, they are potentially good points to split the data into 2 chunks. And this is basically what I'm doing. Then after you split this set of cells, this data, into 2 chunks, you can also divide the genes into the genes that are more specific or most relevant to the first chunk, or in the left, or the second chunks on the right. And what you get is basically a group of cells, again, on the column, and a group of relevant genes, and a second group of cells with a second group of relevant genes. And this is the basic step of the backspin algorithm. And now what we are going to do is just to repeat this. So after the first split, we get 2 chunks. We can take each one of them and repeat the same procedure again. So sort, find breakpoint, and split. Okay? And this is how it will look after the first split, and this is how it will look after the second split. Basically, we will repeat this until we get to the number of splits requested by the user. So this is a parameter. So the maximum number of clusters possible is 2 to the power of number of splits. But usually it will be less than this, because if the algorithm cannot find more variability, it will just not split the cluster. So after the first split, after the second split, after the third split, etc. And now I show one small example on a toy data. So this is a toy data that we generated by Joel Elamano in the lab. And it illustrates what happens if you just apply the spin algorithm. So if you just apply the spin algorithm, spin knows very nicely to sort the cells and the genes into a great one-dimensional order, but it cannot find those borders between the groups, between these clusters. So it's not an algorithm that can identify these kind of blocks. So this is where backspin can come into action. And this is how it looks, for example, after 2 splits. And you can see that, for example, in 2 splits, we were supposed to get up to 4 clusters, but actually we see here 3 clusters, because this cluster, for example, cannot be splitted anymore. So the algorithm just stopped splitting it. While if we now are going to continue and do more splits in the backspin, for example, after 6 splits, this is the picture that we are going to see. And as you can appreciate, this is quite good, resembling the groups of cells and group of relevant genes that are basically relevant, even that this matrix contains basically a lot of noise, as you can appreciate on the background here. Yeah, so that's all about the data analysis and the technical protocol, and now I will hand to Gonçalo.
Okay, so now I'll continue from Amit's presentation and give an example of how we have applied the techniques that Amit has talked about, single-cell RNA sequencing, to the identification of cell states within the oligodendrocyte lineage, which is the major cell type that we are interested in my lab. So oligodendrocytes are a glial cell type in the central nervous system that produces myelin, which is this insulating membrane that insulates the axons of neurons and then allows the fast transmission of electrical impulses within the central nervous system. These oligodendrocytes also have function providing metabolic support to neurons. Now, oligodendrocytes arise from neuroepithelial cells, from neurostem cells, about mid-gestation during mouse embryonic development, around embryonic day 12.5. And these neural stem cells originate embryonic oligodendrocyte precursor cells, OPCs, that throughout embryogenesis remain proliferative and only at postnatal stages start differentiating and then producing the myelin. There is a subset of these oligodendrocyte precursor cells that remain in the adult central nervous system, and they constitute around 5% of the cells in the central nervous system, and they're in an almost quiescent state, meaning they are dividing very slowly. However, upon specific stimuli, they can be activated, start proliferating again, and then differentiate and produce myelin. And the one example of this, for instance, is in multiple sclerosis. So multiple sclerosis, there is an autoimmune attack against the myelin, leading to demyelination. And this leads to slower transmission of the electrical impulses in the central nervous system, and then leads to the motorical symptoms that the patients usually have. In the first stages of the disease, the patients have the symptoms, but then have a recovery. And part of this recovery is due to the fact that these oligodendrocyte precursor cells can migrate to the site of lesions and then differentiate and produce myelin, doing then remyelination. However, when the disease becomes in a progressive state, some of these precursors can be still in the lesions, but they fail to differentiate. And this is one of the main questions that we are asking in our lab, is how the epigenetic state of these oligodendrocyte precursor cells correlates with their capacity to differentiate and to myelinate. Now, as I told you that that oligodendrocytes are first specified embryonically, and studies from several groups have shown that there is distinct waves of oligodendrocyte precursor specification, with the first waves occurring both in the spinal cord and in the forebrain around embryonic day 12.5, then a second wave at embryonic day 15.5, and then a third final wave at postnatal stages. And this diversity of origin of the oligodendrocyte precursors then could suggest then that they would originate different kinds of oligodendrocytes and possibly with different functions. Toxin a study done by Bill Richardson's group clearly showed that when you eliminate using diphtheria toxin gene one of specific waves, some of the specific waves of oligodendrocyte precursor cells, as depicted here, the other waves can rescue the phenotype and can then contribute to the normal generation of oligodendrocytes, and the mice have normal behavior, suggesting that these different waves can be redundant, and then meaning that perhaps oligodendrocytes can also be redundant. However, if we go back in history into the 1920s, when oligodendrocytes were described by Pio del Rio Ortega and also later on by Wilder Penfield, and they described using carbon silver impregnation techniques 4 kinds of oligodendrocytes with distinct morphologies, already by that time suggesting that oligodendrocytes could have different, could be heterogeneous. So in order to address this, as you have seen from Amit's presentation, single-cell RNA sequencing is a very powerful technique in order to examine whether indeed there is heterogeneity in the oligodendrocyte lineage. First, in a study that my group was also collaborating, but it was mainly driven by Stan Lindersson's lab and Jens Jørgen-Leffen's lab here at our unit at Karolinska Institutet, and in which Amit and also Anna Manchado in Jens Jørgen-Leffen's lab were the first authors. Basically, single-cell RNA sequencing was done in 2 specific regions of the brain, the somatosensory cortex and also the hippocampus, using the technique that then Amit said in an unbiased way in order to try to 3005 cells were sequenced. And then applying backspin, as Amit described, 9 classes of cells within these 3005 cells were identified, among which different kinds of neurons, astrocytes, oligodendrocytes, and so forth. So this is where our group stepped in, because doing a second level of analysis through backspin, we could detect that there were 6 distinct populations of oligodendrocytes. And here you can see different genes that here every bar correspond to a single cell, and here you can see the different genes that are expressed in the different 6 populations that we have identified. What was also interesting in this analysis was that here in blue you have cells that come from the cortex, and in yellow cells that comes from the hippocampus. And what we could see was that some populations, like this population oligo3, was mainly present in the cortex, but not in the hippocampus, suggesting that there was also some region specificity. And then this prompted us then to extend this study and investigate the oligodendrocyte heterogeneity in 10 different regions of the central nervous system, both in the dorsal to ventral axis and also anterior posterior axis, analyzing a total of 5,072 cells of the oligodendrocyte lineage, again by single cell RNA sequencing, and then do clustering by clustering analysis. This study was again spearheaded by Sueli Marques, a postdoc in my lab, and also Amit in Stan's lab. And in this, then applying backspin and then doing also correlation and gene expression analysis, what we identified was 12 distinct oligodendrocyte cell states with 2 precursor states, 2 newly formed oligodendrocyte states, 2 myelin-forming oligodendrocyte states, and then 6 distinct mature states. What was also interesting was that we identified an additional cell state, which we call VLMC, which expressed PDGFR-alpha, which is one of the canonical markers for oligodendrocyte precursor cells, but however did not belong to the oligodendrocyte lineage. And I'll talk a little bit more in detail later on. So this is another way to visualize the data. This is a Disney plot where basically it's a way to reduce the multidimensionality of the data, and every single cell here is represented as a single dot, and if cells that are similar to each other will be close together in this Disney plot. And what we could find is that the 2 PDGFR-alpha populations were clearly separated from the remaining populations. These VLMCs that does not belong to the oligodendrocyte lineage, but also the oligodendrocyte precursor cells. And then we have a continuum of cells of the oligodendrocyte lineage, starting with precursor cells, newly formed oligodendrocytes, myelin-forming, and then at the end we would have then the 6 mature oligodendrocyte populations. Going more into detail in some of these populations, for instance here into this VLMC, as I told you before, this cell population expressed PDGFR-alpha, a marker of oligodendrocyte precursor, however did not express SOX10, which is a marker of the oligodendrocyte lineage, and expressed other markers such as COL1A2 and others that were more related with the pericyte lineage. These cells also presented some specific markers that are not in the pericyte lineage, like LUM, as you can see here in this single molecule fish, which was performed by Simone Kudeleppi in Sten Linnarsson's lab. Another population that we were very interested in was in these committed precursors and also the newly formed oligodendrocytes, which were expressed genes like the transcription factor TCF7L2 and also ITPR2. And these cells were distinct from oligodendrocyte precursor cells, as we could not detect, as we did immunohistochemistry in juvenile CNS, they were not overlapping with PDGFR alpha, so they were distinct. However, when we did lineage tracing using the PDGFR alpha GFP or PDGFR alpha Cree GFP, we could see that there were cells that were double labeling for GFP and ITPR2, indicating that these cells were definitely progeny of oligodendrocyte precursor cells. In the postnatal, in the cortex and in the brain at postnatal day 21, we saw around 20% of the cells of the oligodendrocyte cells being ITPR2 positives. This percentage was higher at postnatal day 7, where myelination is really starting. But what was also very interesting is that we could detect at the adult mice, also, for instance, in the corpus callosum, around 10% of the SOX10 positive cells being positive from ITPR2, indicating that these newly formed oligodendrocytes are also present in the adult brain, and then suggesting that they might have a function there. So in order to investigate that, what we did was that we started a collaboration with Bill Richardson's lab in the UCL in the UK. And Bill's lab had previously described the complex wheel running paradigm, in which mice are put in a wheel, but some of the steps are removed. And because in order the mice have to learn how to run in this complex wheel, and this process takes some time until they can reach again the normal speed on this complex wheel. So what Bill's lab has shown in his study was that differentiation of myelination from adult OPCs was important for this process of learning in this complex wheel method paradigm. And then in this collaboration with Bill's lab, what we examined was in these mice after 2 days under the complex motor learning paradigm, what was how did the ITPR2 positive population, these newly formed oligodendrocytes and committed precursors, reacted. And what we could see was an increase in 50% of this population, indicating that in this complex motor learning paradigm, this population is indeed increased, and suggesting that it might have then a function in this paradigm. So if we come back to our data, one of the advantages of having done 10 different regions in this single-cell RNA sequencing analysis is that we could then deconvolute the data and see how the different cells that we have identified were present in different regions. And one thing that we have detected was, for instance, that some regions like the striatum and the substantia nigra vitae, we could detect very low numbers or even none of oligodendrocyte precursor cells, which we still don't know if it really reflects the absence of these cells in these regions, or if it is more like a question of a technical question of the method at this point. Another issue that we could detect was that there was a common path. The common path of initial differentiation was present throughout all the 10 regions that we had identified. But we also saw that the mature cells were present in some regions that we call them mature regions, were absent in other regions like amygdala and the hippocampus that are then more immature in this juvenile stage. And then we had some other regions that were like the hypothalamus, corpus callosum, striatum, and zona incerta, that were regions where we could detect both cells in both early stages and late stages. So what we call an intermediate stage of the oligodendrocyte lineage in the juvenile brain. Another way that we can analyze the data is then seeing the different populations that we have identified, how they are enriched in the different regions that we have examined. And what we could see, for instance, was that some of the mature population seem to be particularly enriched in some of the regions that we have identified, suggesting that distinct mature oligodendrocyte types are then present enriched in specific regions. We could also, in this data set, we not only did a single-cell RNA sequencing in the juvenile mice, but we also did in the adult cortex and corpus callosum. And with this, we could also compare, do a temporal, apart from the spatial comparison that I showed you before, also a temporal comparison of this of the different populations. And what we could see, for instance, was that oligodendrocyte precursor cells in the juvenile mice, the cells are labeled here in red, while in the adult mice are labeled in blue. And we could see that there was not much of a difference between the transcriptional profiles of the juvenile and adult OPCs in between into these different stages, except at the cell cycle level, where we could see that, for instance, cells in the juvenile mice had a higher number of cell cycle genes being expressed in comparison with cells OPCs from the adult mice, which then correlates with data in the literature, for instance, from the Bill Richardson's lab, that indicates that the cell cycle time of OPCs in juvenile mice is faster than in cells that are in the adult mice. And another thing that we could also detect was that, for instance, the mature states of oligodendrocytes, that some states appeared in the juvenile mice, but then appeared to disappear for instance, in the cortex, in the adult cortex. While some other cell types appeared mainly in the adult mice, suggesting then that perhaps some of these mature types might be achieved already at the juvenile state, but might be in some cases transient, while other of the mature states can only be achieved in the adult brain. So, of course, having this diversity of these 6 different types of mature oligodendrocytes, one question that would be quite interesting to follow then is, do they have different functions? And we have started assessing this by doing gene ontology analysis of this data set. And one thing that we could see is that in these mature populations, they all express genes involved in myelination. However, a subset of them, for instance, MOL5 and MOL6, that are present more in the adult corpus callosum and cortex, they present genes, they are enriching genes involved in synapses. And one example of this is the metabotropic glutamate receptor GRM3. We then did an analysis of other glutamate receptors and other channels expressed in our data set, and what we could see is that the different populations express this receptor, although many of them express it in a scattered way. So applying single-cell RNA sequencing, as Amit explained in the first part of this webinar, allowed us then to dissect the heterogeneity of the oligodendrocyte lineage in the central nervous system, both in the juvenile mice and also in the adult mice. And from this data set, the conclusions that we took is that, firstly, ubiquitous of the CNS is the first populations that we find in the initial steps of differentiation, mainly the oligodendrocyte precursor cell, the committed precursor, then the newly formed oligodendrocytes 1 and 2, and then the myelin-forming 1 and 2. But then we appear to see a region specificity when it comes to the mature oligodendrocyte populations, with some of these cell states being present in some regions more than in others. And another aspect is also that some of these populations seem to be present more in the adult versus than the juvenile brain, indicating also a temporal aspect in the distribution of these mature oligodendrocyte populations. So with this, we would like to acknowledge the people that have contributed to the work in this that we have presented. I mean, in my lab and also the Linnaean lab where Amit is, and also several other labs within Karolinska Institutet, and specifically in our unit that have contributed to these studies, namely the Jaring first lab, Enfors, Arenas, and Arsenis lab. Also to Bill Richardson, that we have collaborated for the complex wheel motor learning paradigm, and also the funding agencies that have funded my lab, and also SENS lab, and other labs that have funded this study. So with this, I would like to give the word to Davide, and then me and Amit will be, after Davide's presentation, will be here to answer any questions that you might have. Davide?
Okay. Thanks, Amit and Gonçalo. I would now like to tell you about some of the products and resources that we have at APCAM that can help you with your RNA and oligodendrocyte research. Many of you will be aware of our range of products. We have several antibodies to oligodendrocyte markers. Our key antibodies against markers for mature oligodendrocytes are oligodendrocyte-specific protein antibody, rated 5-star and referenced in 10 papers, myelin basic protein antibody, rated 5-star and referenced in 17 papers, myelin oligodendrocyte glycoprotein antibody, rated 5-star and referenced in 3 papers, OLIG2 antibody, also rated 5-star, and SOX10 antibody, referenced in 3 papers. We test oligodendrocyte marker antibodies to make sure that they work in IHC and ICC, as well as other applications. You can find out how other researchers have used these antibodies by looking at the user reviews and the publications listed on each of the product data sheets on our website. I'd like to draw your attention to the OLIG2 and SOX10 antibodies for a moment, which are labeled RAPMAB. These are rabbit monoclonal antibodies and are ideal for staining mouse or rat tissues versus mouse monoclonal antibodies. As featured in the science paper from today's speakers, different types of mature oligodendrocytes are enriched for particular markers. For example, we have seen how metabotropic glutamate receptor 3, GRN3, labels MOL6 oligodendrocytes. In the paper, the authors confirmed this by IHC using our anti-metabotropic glutamate receptor 3 antibody, AB140741. In addition to markers for mature oligodendrocytes, we also cover markers for oligodendrocyte precursor cells. Here are 2 examples: PDGF receptor alpha antibody, rated 5-star, and NG2 antibody, rated 4.5 stars, and referenced in 6 papers. These 2 are tested to ensure they work in IHC and ICC. In addition, we have many resources around neuronal markers and IHC on our website. These include, for example, a guide to neuronal markers, a specific one on Schwann cell markers and their functions, a glia and demyelinating poster, and an IHC application guide. We also have many resources around RNA and epigenetics on our website, which you can find all in one place on the epigenetics portal at apcam.com/epigenetics. Here you can find useful information on applications such as RNA immunoprecipitation, microRNA profiling, and chip sequencing. thank you for listening, and I will now hand you to my colleague Jessica to start the Q&A session. Okay, so we have a question from our audience on the RNA-Seq technology. So this is probably for Amit. The question is, are there available methods for single-cell RNA-Seq analysis that would include poly-A- RNAs?
So by and large, I think the answer is no. So majority of the protocols that are available today and that people use are using poly-T primer for the reverse transcription. But I think there are people that I know working on it. I don't know if it's published or not. MicroRNA in single cell. I think even Fluidi maybe have a kit for microRNA, and some people that do RNA from nuclear RNA, and then they need to use some kind of random primers to start the reverse transcription. But again, the problem is how to avoid the ribosomal RNA when you do it in single cell and you cannot make any depletion.
Okay. We have a second question from the audience, and this is on backspin. So this is probably for Amit again. And the question is, in backspin, when should the splitting of the clusters stop?
Yeah. Okay. So yeah, of course, this is the million-dollar question of the whole field. What is the So first, whether there is a general working stopping condition, and how many clusters one would expect. And I don't think, again, that there is a very clear answer to this question. It doesn't matter which clustering method you use. There is always, you need to tune the parameters and basically run the methods with different parameters and see where things make sense. Particularly for backspin, basically the main parameter is how many splits to do. And again, this is very different depending on the cells you analyze. And what we do as a practical guide, what I can say is that what you can do, you can always try to do a larger number of splits, and either sometimes the algorithm can identify that there is no need to split more, and this is the good case. And in other cases, it will still split, but then you need to evaluate the clustering results and inspect them manually. And this is basically what we usually do as a standard practice. We run the backspin. And again, depending on the data, you need to have a little bit of feeling of how many splits you should ask. And then you should anyway evaluate the result of the clustering by looking whether two clusters are really different and have any kind of difference. And what we usually do, in case that there is no difference, we can merge the clusters. So the rule in this kind of unsupervised analysis is that we don't create the clusters based on markers. We don't create the clusters in any arbitrary way based on previous knowledge. We let the algorithm do it. The only thing we can do is to merge clusters after they already created. Yes.
Okay. Thanks, Amit. We have a third question from the floor, and this, I see, might be for Gonçalo. It's on oligodendrocyte populations. So what do you think are the functions of the different oligodendrocyte populations you have identified?
Okay, so this is something like I told in the presentation that we've been starting to address first by gene ontology, what the different populations, what genes they are enriched in, and that whether they can give us like a clue on which function they might exert. But one thing that we are actively doing now in the lab is correlating some of these markers that we have identified, and so collecting the transcriptional profiles with immunohistochemistry, so we can be able to see within the brain and the spinal cord where the cells are, their morphology, and then from that point on try to investigate what kind of neurons they might be interacting and what kind of function they might be having. So one thing that I mentioned also was during the webinar was that some of the populations like MOL5 and 6 were enriched in genes associated with synapse communication and so forth. And there was last year, I think it was last year, a paper from Peter Steyer's lab in Canada where he developed the concept of axomyelinic synapse. And I think that perhaps some of the functions of these different populations might be related with this axon-myelin communication. Yes, so David, yeah, that was my answer.
Yeah. We have an additional question from the floor. This might be for you, Gonçalo, again. It's on multiple sclerosis. So you mentioned multiple sclerosis in the beginning of the talk. Are the identified oligodendrocyte populations present and affected in MS lesions?
Okay, so this is our studies that we have done so far, as you saw in the webinar, were mainly in the juvenile and adult healthy mice, and what we are now really focusing in our lab is again investigating by single-cell RNA sequencing the oligodendrocyte lineage in the context of disease. So this is something that we are very much interested, and we think that this kind of technique can give us very useful insights because, for instance, one of the populations that we identified, this ITPR2 positive population, we think screening the literature, it had been previously shown that lesions of MS patients have OPCs, that a population of oligodendrocyte precursors that very paradoxically are Ki67 negative, so they are not proliferating. And they have other markers like NKX2.2. And when going back to our data set, these are very similar to, for these committed precursors or newly formed oligodendrocyte populations that we have identified that are ITPR2 positive. So we think actually that those populations in the lesion might be these new populations that we have identified, and so this is why we think it's quite interesting to examine this now in the context of disease.
Okay, thanks. There's another question from the floor, and the question is, what are the criteria to design or prepare UMI? Who wants to take this?
Yeah, I will take it, Amit. So basically, UMI is a general name for a set of random nucleotides. It really depends where do you use the UMI, but basically we have it on the template switch oligo. So it depends if your template switch oligo is made out of DNA or RNA or some special nucleotides. In our case, it's an RNA oligo. So there is no special criteria except the size of the number of nucleotides inside this set of random nucleotides. And of course, the longer the UMI, you can encode larger number of molecules, like more and more molecules, because it's just a number of combinations that you can get from a set of random nucleotides. In our case, it's 6, so it's up to almost 4,000 combinations that are possible. And for this application, it's more than enough. People use UMI also in a 3' protocol. So the protocol I described is a 5' protocol. People are now using a lot of 3' protocol where they use the UMI on the 3' side, on the poly-T, and then they need longer UMI, and they use UMI which is up to 10. But again, the UMI itself has nothing special. It's just oligonucleotides that you buy from one of the standard companies without anything special. That's all.
Okay. So the next question is on single-cell isolation. Is it possible to explain how the single cells are isolated?
Yes. So again, in this study, what I described is basically the use of the Fluidigm platform. The Fluidigm platform, again, it's a microfluidics platform that basically the cells are flowing in a channel, and this channel has, I will try to highlight, I don't know if people see this capture site. Basically, the cells are flowing through a channel, and the channel has a capture site, which is just some geometrical shape that cells tend to stuck there. And once they are stuck in this capture site, again, it's designed in a way that more cells are less likely to get stuck on the same capture site. So basically, the cells are not isolated one by one. What you do, you load into this fluidic basically a cell suspension, which is basically after you dissociate the tissue, you have a cell suspension with a concentration of a few hundred cells per microliter. And this is, you load about 5 to 10 microliters into the chip, and the machine basically push those cells into the microfluidics channel until you, and this process takes about 30 minutes. And then you go and you image this chip, and you see how many, if the cells got captured or didn't got captured, or how many got captured.
I mean, I could add perhaps that, I mean, before this isolation step, there is also the extraction step. And this is like one of the key things in the single-cell RNA sequencing, because it will affect what cell you have represented when you are doing the analysis. So it's actually a step that in both these papers that we have presented took a lot of optimization in order to get the cells in the right state in order to be able to do single-cell RNA sequencing. I mean, we are working with the brain and spinal cord, which have a lot of myelin, and it's particularly difficult. So depending on your tissue of interest, it might be easier or not to do this extraction step.
So there is indeed one more question from the floor, and it is, what is other information that can be derived from RNA sequencing other than gene expression?
So basically, I mean, the measurement is gene expression. That's all. But of course, gene expression is, again, contain a lot of information. Again, in this kind of platform, and again, this is just one example of platform for single-cell RNA-seq, one can also image the cells with standard epifluorescence microscope, so you can get basically the bright field that tells you the shape and the size, and any other fluorescence channel. But other than this, today there are other protocols, not the protocol that I described, that can do basically DNA sequencing and RNA from the same cell, DNA and methylation, RNA and methylation, and other combinations of protocols that can basically take measurements on a single cell and get more than just the RNA.
Yes, I mean, I would just say that, of course, when you get these gene expression profiles in the single-cell level, you can then try to deduce what kind of transcriptional networks are operating in the individual cells and what kind of transcription factors are driving. But this is all analysis that would be predictive, and then you would need to go to techniques like Amit has referred in order to confirm that those chromatin states that allow the gene expression are indeed establishing those kind of cells. And so, like Amit had said, DNA methylation sequencing in single cell has been developed, but also like ATAC-seq in single cell has also been published recently. So I think it's a start. We can see the gene expression analysis as a starting point for other investigations. And like in our case, in our lab, we are very interested in epigenetics, and I think this is a really good tool to have.